how many hydronium ions are in an E. coli cell. And we had several options. One was 10 to the 2, 10 to the 2 to the 1 to 10 to the 3, 10 to the 3 to 10 to the 5, 10 to the 5 to 10 to the 7, and 10 to the 7 to 10 to the 9. The question is kind of clear. And hydronium ions, H plus or H3, H3O plus, useful in many, many reactions. And we like to do survey. So who's voting for A? 1, thank you. Who's voting for B? 4 people. Who's voting for C? The majority, 100% with one significant digit. And a few are voting for D. So we're missing something in order to answer it, right? There's one missing piece in order to be able to use what you have. What is the missing piece? Yeah, you need the concentration, right? Where would you get the concentration?
Acidity.
Yeah, the acidity or pH, right? Which is what? 7. It's not 10. We could make it 10, but it's 7. So the pH.
Here it's important because it's a log scale, right?
Yeah, you're right. Yeah, here it makes a difference between 7 and 10, but this is like a crazy situation where you're working in log scale. So pH of 7, what does that mean? pH is defined as the minus log of the concentration. Okay, I think it's like literally, I think like H is like the H plus if you want. P is like the something with the log, and there's a minus, so that's like the minus log of the concentration of H plus. That's equal to 7, which means that the concentration of H plus is 10 to the minus 7 molars.
The P is for power.
Power, great. So P is power, which is like, the power is like the exponent of 10. I love it. So it's the power of hydrogens, and somehow it's a minus. I don't know, they didn't want minuses, so it's like, okay, so we have 10 to the minus 7 molar. Okay, can you help me? How do we get from here into the answer for this question? Remember, we had like a rule of thumb, how to go from concentrations into absolute numbers in the molecular world, in the microscopic world. Right, so we had the rule of thumb was 1 molecule, 1 nanomolar. Right, molecule, bacterial cell, you're right. Oh, bacterial volume, actually. It's actually bacterial volume is a better name because I want to move between I don't care about cells so much. I want to talk about like sizes, like volumes, which are my units, this 1 micrometer cubed. Okay, so how do I bring it together?
You have 10 to the power of minus 7 molar.
Okay, so 10 to the minus 7 molar. Let's go to nanomolar. How much would that be? So that would be like this, let's multiply by 1, right? It's 1, the molar needs to be at the top, so I want to have 1 molar at the bottom, and I want to have nanomolar, so that's 10 to the 9. Right, I multiplied again by 1. You can see I got confused for a second. Why? Because I was putting the molar at the top, but no, I need to get rid of the molar. So I'm getting that I have here 10 to the 2 nanomolar. So we're very quiet now, right? Why are we so quiet? Because it sounds surprising, right? Like the math led us into saying, oh, it's 100 nanomolars. 1 nanomolar is 1 molecule. So 100 molecules, or 100 ions in this case. So this is the answer.
But isn't the question about mammalian cell?
No, the bacterial. No.
This was in an E. coli cell. Okay, so now we're moving into trying to find also excuses for ourselves, whether it makes sense or not. Who knows what's happening here? But we did the math, and let's say before we talk about the biological aspects, etc. I just want to show you again. I hope if I'm not convinced you now, I would never convince you. It's a useful thing, right? Like this one rule of thumb enables you to know about water, about pH, about P53, about your protein of interest. It really enables you to go from one thing to another pretty easily without needing to every time do the calculation, which by itself probably you would never do because you would be afraid of like just dealing with this thing, even though it's only multiplying by 1 a few times. Okay, so.
But isn't it really surprising? It's only 100 molecules.
Yes, it is. That's why I chose it. No, it's really I think it's super interesting. There's a lot to discuss if you want. I was corresponding about this with some people studying those things. I think one thing that helps, think about it is these things also diffuse very fast. So you can talk about how long does it take until it reaches, if you have some catalysis of some reaction, it actually reaches that very quickly. That's one thing. And then there's also the lifetime. They have very short lifetime, meaning that like they annihilate, like this every H plus meets OH minus. They diffuse quickly, and then they reappear somewhere else. Not themselves, but like some other two water molecules turn into H3O plus, which means that at every different location, if you need this, it actually appears all the time. All the time we could do the calculation. And that means that like actually it's available, but just for short lifetime. But in that short lifetime, we could already do the reaction. So there's all sorts of things to be said about how this still enables us to be alive and kicking, even though it's a small number. But I think it also shows like it clearly raises many questions you would never do. Like we were all very happy with knowing pH is 7 and live our life without any worry, right? But now when we know that pH 7 has a meaning with absolute units, now we became curious about it, and it raises new scientific questions. And I think that's the utility.